Hi everyone, I'm Gaia. I'm a postdoc in Edinburgh in Barry McCall's lab, and I work in stroke. So particularly we're interested in understanding the consequences of ischemic stroke in relation to how someone might develop dementia following stroke. And we are particularly interested in understanding the role of resident cells in the brain, like microglia, and distinguishing them from cells that infiltrate the brain following stroke, such as peripheral myeloid cells, such as monocytes and monocyte-derived macrophages. And the issue so far in the field to try and untangle what cell does what. These guys, when they infiltrate the brain, they become phenotypically similar to these guys. So it's very hard for scientists to tell these two populations apart. But we know that they do different roles in disease, and that might also help us understand how you have a good stroke recovery. And if people don't have a good stroke recovery and have dementia, it might be because one of these two cells might be playing a role in that. So to get around that and to try and address what these guys do and what these guys do, I've used a novel mouse model, which is called the Hemispheric Cree Rosa TDT. And this is a fancy reporter model that basically lets me label these cells because they originate from the bone marrow. They'll be labeled with a fluorescent tag, which is TDT. But because microglia that are in the brain don't originate from the bone marrow, they will not be labeled with TDT. So once they're in the brain, because I can do a surgery to mimic stroke, which is called DMCAO, I can then tell apart what is a microglia, so what was already in the brain, and what has been infiltrated into the brain following stroke. So with this model, we firstly understood that at acute time points following stroke, so at 3 days and at 14 days, the TDT positive cells only are in the site of the stroke. They don't actually go anywhere else in the brain. And we were interested in that because we think that if you develop cognitive impairment or dementia in stroke, it might be that the brain is changing across and not just at the site of the stroke. But we understood that once these guys infiltrate the brain, they only stay within the stroke. So they have a very localized action, basically, to helping out, mopping up all the stuff that has been degenerating following the stroke. Whereas microglia, if we go down here, so the way that we've done this is through something called single cell sequencing. So we're actually looking at the individual role of each cells within these areas of the brain. And we actually looked at microglia following the monocytes. And we found out that these guys become activated, or they're responding to something across the whole brain. And that these guys remain angry or remain activated even 3 months following stroke. So there's really something to do with how chronic their response is that we think is driving possibly the development of dementia, because it's affecting the whole of the brain, brain-wide. And I'll leave it at that.